I'd like to start by thanking the organizers as well for putting together what I think is a very important program, and I'm delighted to be a part of that and look very much forward to the discussion over the next 2 days, because I think we're all of us who are speaking or sitting in the audience are clearly struggling with this issue of how to account for these cumulative impacts in children's environmental health. So I'm going to start with a map. So I was asked to talk about some of the work we've been doing to look at cumulative environmental toxic effects on particularly urban asthma and lung growth and development in some of our populations. And this is what motivated me early on in my career when I was a pulmonary fellow, looking at the rising trends in asthma and seeing it disproportionately burdening lower income ethnic minority populations, particularly in the inner city. And this is a smaller scale than what you were just seeing for California, but this is the Boston metropolitan area. And again, as you go darker, you see increased asthma morbidity, looking at asthma hospitalization rates in this case as well. Also, those are the communities where they're lower SES. They're primarily ethnic minority. They're the ones that are being particularly impacted. And so one of the things we were interested in is developing studies in epidemiology where we could actually start to unpack that. Well, why is there disproportionate burden there? So traditional risk factors, certainly looking at family history, heredity, low birth weight, maternal smoking during pregnancy. People were already looking at the physical toxins, ambient pollutant, indoor allergens, and also risk for infections. But really those explain some, but relatively little about what was going on in the low SES and ethnically mixed populations. And what we all started thinking about and talking about was differential exposure to these environmental factors. So folks who live in inner city environments where they're more perhaps polluted may also be at increased risk of experiencing greater amounts of psychological stress. And that's really what we started to look at. How do these things co-vary? What are the independent contributions of the psychosocial factors? And also how might the psychosocial factors even enhance the effects of the other environmental toxins? So looking more at the cumulative risk. These aren't new ideas, just to sort of let you know. We're not the first currently to think that psychological problems have a role to play in asthma. You can go back all the way to the 12th century. There's a treatise written on the link between the mind and the body related to asthma. Actually, as early as the middle part of the last century, asthma was really thought to be something that was in your head. It was one of the 7 psychosomatic illnesses called asthma nervosa. And then we sort of took a shift. We understood there's a huge component of asthma that's related to allergy, and we all started looking at the biology and the immunology of asthma. And really what we're trying to do now is try to bring those things back together. And this is shown here in this cartoon. There's a lot of evidence for the link between mind-body. Sort of anecdotally, we all understand it in ourselves. When we experience stress, we're more likely to get sick in different ways. And there's a lot of biology to show the connections between the brain where you emotionally experience and respond to the stressors that you're facing. And there's release of neurohormones and neurotransmitters that go systemically and can have broad health effects, including on the lung. And this cartoon just shows that there's a lot of evidence to suggest there's strong links in back and forth talk between the brain and the lung, through the immune system, through the autonomic nervous system, through the hypothalamic pituitary adrenal axis and neurohormonal systems, and connections with cortisol exposures and those sorts of things. And those are the things that we try to measure in our work to see if we can actually show these mechanisms are operating. And then to think that, well, what is health? It's an optimal balance of these key regulatory systems. And there's a lot of work in animal studies as well as human data now that suggests that in a lot of the environmental toxins that we talk about, whether we think about psychosocial stressors or many of the physical toxins like air pollutants, tobacco smoke, allergen exposures, they're all pushing on these systems. These key regulatory systems in the body like to live in some optimal balance, and they're all talking back and forth to each other, so you can see how it starts to get complex. And when those are shifted over time, when you have chronic exposure to these environmental toxins, you start to see adverse health effects. So I just put that picture there because that's kind of what we try to keep in mind when we are developing our studies. So, well, we're all under stress. Why aren't we all sick? Well, stress in and of itself isn't necessarily bad, and we all need a little bit of motivation in our lives. So too little stress can actually also be have adverse effects. And there's some optimal level that each of us have our tolerance level, our own ability to cope with. And it's when you're in a situation where you tip over, you're no longer able to cope, there are just too many stressors, they're cumulative, ongoing, they just never go away, and you're living with them chronically. That's where you start to see adverse effects, and you start to see shifts in these key regulatory systems and this imbalance. And this slide just says, well, we know that's true for stress. Stress can disrupt those pathways. But boy, if you get into the literature and you start to understand the science in these other fields of environmental health, you can see shifts in these same systems that some overlap directly with how stress is operating. Some do these through different mechanisms. Tobacco smoke, ambient pollutants, allergens, they're pushing on those same systems. And so you can start to see additive or cumulative effects when you have co-exposures. So this is sort of the broad framework that we work in. So exposure 1 is present, usually exposure 2 is present. They co-vary. Exposure 1 and 2 may co-vary and be one's a proxy for the other, and you only see effects of one. Maybe you don't see the effects of the other, it's just merely a proxy. And then, are there interactions? Are there additive effects? If you see these co-vary together, children are exposed to exposure 1 and 2, you see effects that look like 1 plus 2. Exposures 1 and 2, you might have a multiplicative effect, that they're synergistic effects. Maybe they're operating, pushing on those systems through a little bit different pathways, and you start to see synergistic effects of those. This slide just shows you that what I'm going to talk about today and where I've focused most of my work is really at the early phase of the natural history of asthma. I'm interested in, do these things interact at the very beginnings, the root of the onset of asthma? And so I look really starting in early life, starting even in pregnancy. And you can see that the effects that these have on mothers during pregnancy and during fetal development already start to push and program those regulatory systems in the kids. Their immune system gets pushed towards an asthma phenotype, for example, and you start to see risk that early on. And just as we have science to show that in physical toxins, like if mom is a smoker during pregnancy, that gets to the fetus and has impacts on the lung growth and development and risk for asthma. So does psychosocial stress and can have those same disruptions. So we started a program at Harvard when I was there called the Asthma Coalition on Community, Environment, and Social Stress, the ACCESS project. And this has been now a series of prenatal cohort studies that have been designed to understand sort of cumulative effects of what we started looking at here as the key environmental factors. Stress was the new aspect of it, and we go after that in great detail, very comprehensively, and I'll talk about some of that as we go along. We look at air pollution, so we're looking at mostly ambient traffic-related air pollution, do some work on the indoor environment as well, tobacco smoke. We go into the homes and we measure their allergen exposures, because people who live in areas where there's high levels of ambient air pollution, traffic-related air pollution, high levels of psychosocial stress may also live in dilapidated housing and more likely be exposed to things like cockroach allergen, mouse antigen, those sorts of things that we know are important for asthma. And then we look at dietary exposures as well. And in the project that we're going to talk about today, we had 955 pregnant women recruited into the project, mostly from that inner city area where I showed on the map before, through the WIC centers and the participating community health centers, mostly ethnic minority, 50% Hispanic, 35% African American, and primarily low SES. And just to show you, we did a pretty good job of getting a distribution across that area where we were interested in trying to explain these disproportionate asthma burden. You can see that they overlap pretty well. This is just the population that we recruited. Well, the types of stressors, just to give you a sense, we do go after a broad range, and we're finding that particular ones are very predictive of what we're looking at. We do look at recent life experiences like job loss, those sorts of things. Remote life experiences turn out to be very important. A lot of these people have chronic histories of stress, either living in poverty over their lifetime prior to this pregnancy that we're interested in looking at. A lot of these women have a history of traumatic stressors in their lives coming into it, and those turn out to be very important. And then we look at other things. Persistent life difficulties, role strains, racism, discrimination turns out to be a very important one. And community level strains, not only what's going on in the individuals and the family level, but we found that actually what's going on in their community, and one of the ways we measure that is crime and violence exposure in those communities. So very broad look at where these stressors are coming from. Here again, one of the ways we utilize those scores across multiple domains is just to add up across how many domains are they experiencing these stressors. And in the inner city, in these low-income populations, they're experiencing many of these across all of these domains. And that's, again, this idea of we're just pushing them over. It's chronic, it's ongoing, and there's too many of them. And they're going to be more apt to have adverse health effects. And this is just a life course diagram showing, we're looking at all those exposures that we showed in the access slide, and start to look at, well, does it disrupt these mechanisms in pregnancy? And we've actually found some data that suggests that it is getting into the body of these women. It is disrupting and pushing these key regulatory systems, shifting them towards what are likely to translate into adverse health. So one of the things we looked at, well, does it disrupt maternal cortisol? Because we know there's interaction between mom's HPA axis, cortisol floating around, interacting with the fetal placental unit, and that already may start to program the child's functioning of these different key regulatory systems and start to program risk. We show that higher level stress, I'll just point you to this side of the slide, higher level stress in these women turn into looking like this flattened curve here in the red. We actually also looked at this in relationship to who goes on then to give birth to children who have an early phenotype of asthma. In this case, we're looking at repeated wheeze over the first 2 years of life. And in the red are the ones, if your mom had this type of HPA profile or cortisol expression in utero during pregnancy, this is what the child is seeing. They also were the ones who were more likely to go on and have children who had early asthma markers. We also show that it can disrupt immune function already starting during pregnancy, and already there's an imbalance and a shift towards a profile that we think, based on other evidence that we have in the asthma field, puts them at risk for going on to develop asthma as well. This just shows you, again, stress across that multiple domain measure that we do, increasing here on this axis. There's a linear association between the level of stress mom reports during pregnancy and having the odds of having a child with repeated wheeze, which is an early indicator of asthma. It looks the same if you look at it in the first 2 years of life, mom's exposure during the first 2 years of life. Actually, the kids that are most at risk are the ones where there's high stress in utero as well as continuing in that first early part of childhood as well, which I won't show, but just mention. That same increase from low to high level stress across that domain measure is also linear related to some immune biomarkers for both innate and adaptive immunity in the child. So we isolate blood when the child is born in the cord blood, and we're already starting to see differential functioning of the child's immune system when we send them off to the immunology lab and stimulate them with either nonspecific or specific allergen exposures. And it seems very related to the levels of stress that we're seeing as well. Well, what about interaction? So one of the things that suggests there does seem to be an independent effect of the stress, and I should say in all of those models, when we look at the stress, we control for all of those other environmental factors that we know are covariating to some extent and are potential confounders. So we treat them as confounders and say, yes, there's an independent effect of stress. Well, we talked about early on, it makes sense to think about there actually may be additive or synergistic effects if you combine these exposures in these critical periods of development. So in this paper, we looked at interactions between the level of allergens that we measure in the home when we go in and collect dust, quantify how much allergen mom's actually breathing in during the pregnancy, and we looked at that in relationship to the cumulative stress measure. And again, the cumulative stress measure here increasing across this axis, and what I want to direct you to is this line going up. So if there's high levels of stress and high levels, in this case, dust mite exposure, you actually see greater expression of IgE, immunoglobulin E, which again in the cord blood is a marker when you have very high levels of a child that's going to be going on with greater risk of developing asthma. So already we're seeing a phenotype, an immune phenotype at birth that seems to be related synergistically to high levels of allergen exposure in utero, as well as the stress in moms. What about after birth? It's not all over. It starts in pregnancy. I said maybe there's cumulative effects if they continue to be exposed here. And we also looked at the effects of stress postnatally in the families, and particularly in the caregivers, because one of the ways the developmental psychologists have shown that you can measure stress at that early phase is to look at it in the caregiver and provider, because your primary caregiver is very important for that child for attachment and development of their own stress regulatory and emotion regulation systems, and that's what's going to translate to shifts in those key regulatory systems that we have pictured here. So we look at stress in the caregivers and how that might relate to these outcomes in the children. Early on, we showed that there was a direct relationship between a continuous measure of caregiver stress, just moms reporting every couple of months how out of control they felt that their lives were, this a stress appraisal measure, the perceived stress scale. And we showed a dose-response relationship between higher level stress in the caregivers and greater increased likelihood of having that child go on and develop repeated wheeze prospectively as we followed these children. In those same children, we isolated blood when they were about 3 years old. Remember, in the cord blood, we said prenatal stress was linearly associated with disruption, immune profiles that suggested increased risk of an asthma phenotype. Similarly, we did it when these kids were older, and just showing in the white area here, this is mean maternal stress, repeatedly measured over the first 3 years of the child's life. And it looked at lymphocyte proliferative response. How activated were this kid's lymphocytes when we isolated them at age 3 years if we were then to stimulate them with different things, in this case, dust mite allergen. And kids who grow up in these high ongoing homes with ongoing high levels of stress, measured here through caregiver stress, were much more prone to having a very active immune system that was responding to these other environmental factors that they're also seeing during that time. So the stress is sort of priming the pump, and then if they're at the same time being bombarded by high levels of pollutants and allergens and other things that are going to be shifting these systems, we're going to see greater impact. This is busy. I couldn't figure out how to do it in a picture, but I just want to show the other thing that we looked at in these data are interactions with the traffic-related air pollutants. And in this case, we use land use regression modeling to look at black carbon as well as PM 2.5 as our indicators of the air pollutants. We put them in the same models with a community-level stressor here. So now we're not looking at individual level stress across those domains, but I mentioned that another way we look at it is at the community level, and community level exposure to violence and crime levels in the communities is how we're measuring that. And we look at that low, medium, and high levels of community violence, and this is self-reported on a questionnaire. It's not based on institutionalized data. And then, as well as our black carbon and PM 2.5 indicator. Because a lot of people, when we said we were going to look at violence exposure and how that may have an impact on asthma expression in these kids, well, it's just a marker of bad environment. It's going to just be a proxy for bad air pollution. At any rate, what we're showing is that they stay in the model together, and they both have independent effects. You see about a twofold increased risk of having a child to go on and develop repeated wheeze, whether you're in the higher level air pollution or the highest level ECV group. Similarly for PM 2.5. And actually, if you put all three of them in the model, all three of them pretty much stay in there as well, which I show on the next slide, but I won't. I want to sort of talk about the interaction effect. So here's the same model. We've stratified them by their level of community violence exposure. And we're comparing median, above the median ETV, low ETV here, high ETV versus low ETV here in this one. And then we look at it based on low or high levels of the air pollution exposure. We actually, there's suggested a difference. So if you're high level of community violence exposure and low BC exposure, the moderate levels of BC exposure, so below the median, that's where we actually start to see suggestion of an interactive effect. It may be that the effect of the air pollution at the high levels sort of overwhelms our ability to be able to measure above and beyond that what might be going on with the violence. But we do find a suggestion in stratified analysis that there's an interaction. It's not reaching statistical significance, maybe a power issue. We do actually find an additive interaction, a significant additive interaction between the community violence exposure and modest levels of the BC exposure. So people say, well, why isn't it the high group that's most impacted, and is this important? To me, this is important because if we start to do our intervention, we design intervention strategies and we're focused on one thing. This is why cumulative, you got to think of these cumulative risk models, because if you're focused on one thing and you say, I'm going to go in and we're going to try to fix the air pollution issue, and we're going to try to reduce exposure to these traffic-related pollutants, and so you get it down to some more modest level. But yet you don't take care of these other things that are also impacting the effects underlying. You're still going to have risk there if you leave the stress part alone, for example. Similar findings with the PM 2.5, just to show that they're consistent across our air pollution indicators, which we found to be robust. So looking ahead, if these kids are already programmed because of the environmental exposures, these cumulative effects starting in these critical periods of development, as they go along and they continue to be bombarded with these different environmental toxins going forward, that risk is only going to increase as we go ahead. I think we need to, if we're really going to fully understand children's environmental health risk in these low-income, highly impacted communities, we're going to have to start thinking with this sort of model in mind. We're going to have to measure these things together, commit ourselves to doing studies large enough where we can tease this apart and find the evidence of where we need to put our interventions. And this just suggests that, it's not just the individual level. We have to keep all these things in mind, the developmental trends, the critical windows of impact, what are the things operating at these different life stages. It comes from the individual, sort of the maternal fetal and maternal child interactions, the family unit, other social relationships, the communities, and what drives that? What actually leads to high levels of crime and violence in communities? Social disorganization, the amount, it's the social and economic policies that drive that. These things don't just happen. So we have to start thinking very broadly if we're going to actually start to switch up some of these trends. And hopefully some of the work that we've done sort of, contributes to that in some way, but I'll tell you, that's why you need meetings like this and put everybody together who's coming at this from very different levels and all across to all of these different action points. This is the kind of thing that we're going to have to do and think about and put our heads around to really make some impact. And so I'm very excited to be here and be a part of that discussion, and I'll end there. Thanks very much.